backgrounds and lots of expertise, both in precision medicine, disease genomics, and also he's a master of everything related to computers when it comes to machine learning and computational biology. He's also a member of the Broad. He's the principal investigator of the Computer Science and Artificial Intelligence Lab at MIT, and also the director of MIT's Computational Biology Group. His research really does look at multiple different genetic and medical illnesses. He has worked in Alzheimer's disease, obesity, schizophrenia, cardiac disorders, cancers, and immune disorders. But I've gotten to know him best because of his emerging work in the field of Down syndrome. Manolis is one of the core collaborators with the Alana Down Syndrome Center at MIT. Many of you know our Down syndrome program here at MGH has a very good, close working collaboration with our colleagues at MIT, and it's through that capacity I really have appreciated all of the work that Manolis and his team are doing, particularly applying it to people with Down syndrome. He has published widely, and he's also received multiple different awards, including the US Presidential Early Career Award in Science and Engineering, and also many other science awards, including those that have been distributed by President Barack Obama. So thank you, Manolis, for joining us. We're really looking forward to your presentation, and thank you for joining us in this new virtual reality.
Thank you so much for having me. It's a real pleasure to be here. So let me start with my disclosure slide. So I'm a co-founder of a secure AI labs company, which is helping people share data. So to whatever extent data sharing is a bias, this is my bias. So what I'd like to tell you about today is our work trying to translate genetic insights into therapeutic insights and focusing on single cell dissection, but also manipulation of disease circuitry. So why are we so excited about genetics? Because the promise of genetics is to reveal new disease mechanisms, to reveal new target genes, new therapeutics, and ultimately enable a rational, personalized, and precision medicine. How is that possible? If you look at genetic studies over the last few years, they have shown a revolution in the ability to discover genetic loci underlying complex traits. This is one example, looking at the BMI association with a dozen loci in the human genome and reigning supreme above them, the FTO locus associated with obesity. This is the strongest and the first, and by far the largest effect on obesity that our genome has. And when it was discovered, it was heralded as a great path to really new therapeutics for obesity. And therefore, a lot of people started trying to understand how is this locus contributing to obesity. And if you zoom in within this locus, what you find is that there are 89 common variants, all sitting in the first intron and a teeny part of the second intron of the FTO gene. FTO was promptly renamed Fat and Obesity Associated, and a huge search costing probably around $1 billion across industry and academia was launched to understand the function of this gene. Many years later, the search came back empty-handed. No one could actually figure out how this gene leads to obesity. And the reason is that this gene is in fact a red herring. Everybody was so excited that there was a single gene underlying this association. So suddenly people were thrilled because instead of having 5 genes in the locus, there was only 1 gene. And therefore there was a lot of confidence that this is probably the causal gene. But if you look closely, you will notice that none of these genetic variants actually perturbs the FTO gene directly. What these variants instead do is perturb likely non-coding regions within this locus. And that's the challenge of GWAS. In 90% of cases, the hits are actually non-coded. And in this particular case, as I'll mention shortly, we found that the true target gene actually sits 1.2 million nucleotides away from this genetic locus. So we can't just look under the lamppost for finding the root of genetic disease. And across 120,000 genetic loci associated with complex traits, that means that in 90% of cases, we're going to have to look for a possible target gene far away. We're going to have to look for the causal variant in new ways because none of them actually affects protein coding genes in 93% of cases. The cell type of action will be not unknown, and the relevant pathways will be unknown. So that will be the challenge ahead of us as we're trying to translate the tens of thousands of genetic discoveries underlying complex traits into therapeutic insights for how things actually work. So what are the tools that our team is using to address these? Well, we start with genetic bases of disease, whether these are chromosomal abnormalities, common variants, rare variants, inversions, locus duplication, CNVs, etc. We then complement these data using RNA and epigenomic profiles. And that's exactly how we're collaborating with Brian on Down syndrome. We're basically collecting brain post-mortem samples to basically start looking for DNA accessibility, histomodifications, DNA methylation, and of course, RNA transcription differences within the individual cells of these post-mortem brains across both healthy and disease samples in a large number of traits, to then start understanding what are the driver genes, regions, and cell types underlying these complex disorders. And in particular, we're using a large number of computational methods for integrating these data sets and for ultimately inferring the path through which genetic variants act in these intermediate phenotypes and ultimately change disease. And our goal is to then validate a lot of these models, as I'll show you shortly, in human cell cultures and and also mouse models of human disease, to basically validate the causality of the individual variants, the regions, the genes, the cell types of action. And of course, we disseminate the results and start all over again. So here's an example of these dissections. If we start with this locus associated with disease, we can zoom into this locus and basically find what are the individual genetic variants that underlie these disorders, recognize what enhancer regions, what distal regulatory control regions these variants sit in, who are the upstream regulators predicted to control these regions, what are the cell types where these regions are acting, and what are the target genes downstream of each of these enhancer loci. The end result is, instead of having just simply, oh, something goes in here, we know the SNPs, the target genes, the enhancers, the cell types, and the regulators. So how does this all apply to the first slide that I showed you of the FTO locus? This is in collaboration with Melina Klausenzer, our advisor in Germany, Hans Hauner, a large number of collaborators across many institutions. So, and this is what I like to say, a paper about a single bit of information in the human genome. This is about a single nucleotide change that changes this T into a C within the FTO locus. So using these approaches that we talked about, what we ended up doing is dissecting this region of association from 10,000 nucleotides down to a single nucleotide variant, which changes this AT-rich motif bound by the AT-rich interacting domain protein ARID5B. When you change this AT-rich motif into a C, this protein is no longer able to bind. This causes upregulation of IRX3 and IRX5, two genes that are sitting 1.2 million nucleotides downstream and 600,000 nucleotides downstream of this particular SNP. And that in turn changes a massive shift in the metabolism of the fat cells of these individuals. The upregulation of these genes leads to a shift from thermogenesis, namely the process of burning calories in our fat cells, in our beige fat cells and our brown fat cells, to lipogenesis, the process of storing energy in lipids into our white adipocytes, and therefore a shift from lean to obese. Why are we so excited about the circuitry? Because knowing the circuitry means we can actually intervene. We can actually start reversing the disease phenotypes. And in particular, we showed that increasing or decreasing ARID5B, increasing or decreasing the downstream target genes, works like a switch and basically shifts back and forth between lean and obese phenotypes at the cellular level, at the regulatory level, and at the organismal level. Moreover, with a single nucleotide change of C to T and T to C, we were able to again shift between lean and obese phenotypes. by changing the primary cells, the primary adipocytes, from risk and non-risk individuals. Here's that experiment, basically looking at how thermogenesis is completely abolished in the C locus that abolishes, in the C allele that abolishes these AT-rich motif. Changing that single nucleotide into a T restores thermogenesis both basally and in the stimulated conditions to the physiologically normal state in primary adipocytes from risk individuals. And if you look at the downstream effects of those genes, IRX3 and IRX5, in the organismal setting, we did not do a clinical trial, but we went into mice and we downregulated IRX3 using a dominant negative form specifically expressed in adipose tissue. And what we found is that the mice lose 50% of their body weight. They lose completely their fat stores. And if you introduce a high-fat diet, normal mice who are heavier to start with gain weight, but these mice are unable to gain weight. So they lose weight in their sleep, they lose weight when they're awake, they don't exercise more, and they don't eat less. It just completely shifts their metabolic regime exactly as predicted by our computational model. So what I'm going to tell you about today is how do we push this to thousands of loci systematically? How do we build reference epigenomes to predict disease-relevant tissues? How do we combine genetic, epigenetic, and expression variation in the context of disease to understand causal genes, tissues, and the directionality of loci? How do we combine single-cell data, particularly in brain, to understand the cell types where epigenomic and transcriptional variation occurs, and how genetic effects act in a cell type specific way? How do we integrate information across many different phenotypes through the medical records? And how do we integrate information across multiple tissues? And lastly, what kind of technology can we use to carry out such studies very high throughput by dissecting millions of regions in individual experiments? I'm trying to look at the chat window. David, can you please put the meeting code here? All right, so let's dive right in. How do we use reference epigenomes to predict these disease-relevant states? As I mentioned, the vast majority of these genetic variants are non-coding, but the human epigenome varies dramatically across different cell types and tissues. So we launched a large effort as part of the Roadmap Epigenomics project and the ENCODE project to systematically annotate the human epigenome across many different primary tissues, both adult and embryonic, different regions of the brain, and also iPS lines and iPS-derived lines, as well as primary cells from the tissues. And in each of those, we profiled a large number of histone modifications, open chromatin, DNA methylation, and gene expression to understand systematically what are the control regions of the human genome and how do they change from cell type to cell type. The result is quite striking. This basically allows us to now start annotating hundreds of cell types and tissues with specific annotations of transcribed regions based on epigenomic signatures associated with transcription, promoter regions based on epigenomic signatures associated with H3K4 and E3, enhancer regions shown here in orange, and also repressed regions, in this case polycomb repression shown in gray. And these regions are basically the elements that DNA is wrapped around. So if you look at DNA, every 147 base pairs of DNA are wrapped around one histone protein. Sorry, a group of one nucleosome, which is a group of 8 histone proteins, H2A, H2B, H3, H4, each in 2 copies, at least for normal nucleosomes. And each of those histone proteins has a long amino acid tail, which can undergo a large number of post-translational modifications. And this effectively paints the chromatin in different colors, each of which colors has a different role. Promoter regions, which I'm going to be showing in red, play a role in initiating transcription and figuring out the exact locus of RNA polymerase binding and eventually transcription of the gene. Enhancers act in very large distances, as we saw in the case of FTO. They can be more than 1 million nucleotides away from their target gene. Polycomb repressed regions I'm going to be showing in gray, and then transcribed regions I'm going to be showing in green. So looking at these, we can now start seeing this picture and how, for example, this PAX5 gene is repressed in most cell types, except for a handful where it turns on. But beyond simply what gene is what cell type is every gene and regulatory region active in, this kind of information gives us linking information. For example, if I have a non-coding region here sitting between those two genes, I might be wondering, well, what gene might it be regulating? And what I see if I look at the correlation between the enhancer activity of this region and the genes nearby, I see that, well, PAX5 might actually be a target of this region because it turns on exactly in the cell types where this region turns on, enabling us to not only figure out what regions these variants might be active in, but also what genes they might be regulating. We can also start looking genome-wide at the enrichment of genetic variants associated with different disorders and different traits with the annotations of these enhancer regions that are active in different cell types. So if I look at genetic variants associated with height, I find that they localize primarily in non-coding variants active, non-coding enhancers active in stem cells. If I look at genetic variants associated with type 1 diabetes, I see that they localize primarily in immune enhancers. If I look at blood pressure associated variants, they localize in heart enhancers. And if I look at cholesterol variants, they localize in liver enhancers, enabling us to paint effectively a diagonal connecting genetic traits to the tissues where most of their genetic variants appear to be acting in. That allows us to now start looking at dozens of different traits and dozens of different cell types and effectively looking at this picture of where is height acting. And if you look at the genetic variants associated with height, they don't enrich anywhere except for embryonic stem cells. Immune traits, T cells and B cells. Blood pressure, nothing except the left ventricle of the heart, where the blood pressure builds up before it's pumped to the rest of the body. Cholesterol, liver. If you look at fasting glucose-related traits, you see that they enrich specifically in pancreatic islets, consistent with type 2 diabetes biology, as we know. If you look at inflammatory bowel disease, well, you have the inflammatory connection with different immune cells, and also the bowel disease connection with different digestive tissues, again, making total sense. If you look at Alzheimer's, well, we're ready to put some money on the brain and say, well, for sure, we're going to see brain. And this is brain right there. We don't see a single enrichment in Alzheimer's disease. Instead, what we're finding is a very strong enrichment in CD14 plus cells. This is a mark of monocytes, which includes both macrophages in the circulating blood and the microglia, which are the resident immune cells of the brain, suggesting that the genetic variants that predispose us to Alzheimer's might actually be acting predominantly in microglia, and only through that having the devastating effects that we know they have in the neurons. So to look at this further, we actually started sorting cells from the hippocampus and the prefrontal cortex, and then asking specifically, where are these genetic variants localizing between neurons, microglia, and other glial cells? And what we're finding is that this enrichment is very specific to microglia, indicating indeed that the vast majority of the genetic risk is probably acting through immune dysregulation in these microglia, which in turn can have a series of devastating effects across many different cell types. Indeed, we published this back in 2015, looking at early and late stage Alzheimer's and what is the order of changes in both immune processes and neuronal processes over time. And what we found is that immune processes change very early on, whereas neuronal processes only change later on, consistent with first action in the immune cells and later on action in neuronal processes. We have since expanded this work dramatically. We went from 127 epigenomes to 834 epigenomes, which we just posted to iArchive about a month ago. And we also went from 54 enriched traits to 500 enriched traits and 30,000 different SNPs that are now sitting in enriched enhancers, providing a highly specific association with disease across countless traits and dozens of different cell types, giving us precise biological hypotheses on their mechanistic basis. That allows us to now start placing a large number of disorders into the tissue they might be acting in. If you look at brain, for example, we find that intelligence, educational attainment, self-reported math ability, etc., localize in brain, just as schizophrenia, neuroticism, depression, worry, etc. do. In the kidney, a lot of filtering functions, in a lot of different cancer cell lines, a lot of sort of corresponding traits. In blood and T cells and B cells, you see a lot of immune traits and blood traits, but also Alzheimer's localizes here rather than in the brain, as we saw earlier. Similarly, with heart, liver, and so on and so forth, you see a lot of corresponding traits localizing there. Coronary artery disease is a funny one. Instead of having a single enriched tissue, such as heart, we find a large number of enriched tissues. So what could actually be happening here? Well, in contrast to highly oligogenic traits, such as Alzheimer's that localizes, well, primarily in immune and some brain cells. If you look at educational attainment that localizes in brain, and if you look at QT interval that localizes in heart tissues, you basically have these multifactorial traits, such as BMI, that involves many tissues, and health span that involves many tissues. And coronary artery disease in particular is one of the champions with more than a dozen different types of tissues involved, including heart, including liver, including a lot of other tissues. So this gives us the opportunity to now use epigenomics to not just pinpoint one tissue, but to understand the complexity of these polyfactorial disorders across many tissues. And in particular, we can take the genetic variants associated with coronary artery disease and partition them according to the specific enhancer classes that they fall in across these different tissues. And when you do that, you find dramatically different gene ontology processes underlying coronary artery disease lighting up. If you look at liver, for example, the coronary artery disease enhancers that localize in liver, they're enriched in very different sets of processes than the CAD variants that localizes in coronary artery. So very completely different pathways associated with different loci, all of which are associated with CAD. And if you look at these partition, you also find that the comorbidity, namely the co-association of these genetic loci with additional traits, is in fact dramatically different. Cholesterol, for example, is co-associated with CAD only in the liver enhancers, but not in the coronary artery disease enhancers, as opposed to systolic blood pressure, which co-localizes in both. And everything I've showed you so far has been about reference epigenomes to predict these disease-relevant tissues, pretending that all of us in this Zoom meeting have the same epigenome, but in fact we don't. Epigenetic variation is almost as large as genetic variation across individuals and as expression variation across individuals. So what we'd like to do next is combine genetic variation, epigenetic variation, and expression variation in the context of disease to understand the path of these genetic variants. And in particular, what we're trying to do is bridge these extremely polygenic path between individual genetic variants that play only tiny effects individually on disease, but there are thousands of those that jointly contribute to disease, to the intermediate tissues and cell types, epigenetic alterations, transcriptional alterations, and endophenotype alterations that ultimately lead to disease, in order to bridge the gap into much more oligogenic traits, each of which is controlled by only a smaller number, every gene a smaller number of regulatory elements, every regulatory element a smaller number of enhancers, every pathway a smaller number of genes, and every disease only a small number of pathways. The challenge, of course, in drawing these unidirectional arrows is that the unidirectionality is only true when we get out of genetics. Beyond that, all these arrows are in fact bidirectional. Why? Because the disease itself could be acting on gene expression changes that perhaps combat the disease rather than cause the disease, and vice versa. Similarly, the environment might be acting on both disease and intermediate gene expression and epigenomic changes, and therefore the causality might neither be forward nor back, but instead correlation, which does not always imply causation. So we need to develop new statistical methods to overcome this challenge and exploit the unidirectionality of genetic information to infer directionality and causality in all stages of this pathway. To do this, we basically started looking at genetic, epigenomic, and transcriptional variation in the context of Alzheimer's disease, using the ROSMAC cohort that David Bennett has assembled at Rush University, and in collaboration with Phil de Jager, previously at HMS and now in Columbia University. Specifically, we looked at many regions of the brain, and in particular the dorsolateral prefrontal cortex, which is associated with cognition and is greatly affected in function in many disorders, including Alzheimer's disease. And we looked at how genetic variation first allows us to predict the epigenome, and then how together they allow us to establish causality for these disease regions. Here's the first part. How is the genetics of a person affecting the epigenome of a person? What we found across 50,000 different loci is that if I know the genetics of a person at conception, before even birth, I can predict their epigenome at 93 years of age in an inaccessible tissue like brain with very high confidence. If I know that this individual carries these genetic variants, an AA, an AT, or a TT, I can effectively predict their epigenome at this particular location. And that's true across 50,000 different locations across all of these dramatic range and significance. Up here is 10 to the minus 300, down here is 10 to the minus 10, and the genome-wide significance line is the red one shown here. So that basically means that I can predict the epigenome for many of these loci. Why is this exciting? Because I can use that to get causality back. What does that mean? If I look at GWAS, I am trying to go from genetics all the way to disease, hugely polyfactorial trait, as I mentioned earlier, with tiny little effect sizes. That basically means I need tens of thousands of cases and tens of thousands of controls in order to be able to get robust inferences, each of which has a tiny effect. Instead, I can look at MWAS, methylome-wide association studies. That is a correlation between methylation changes and disease. This is a more oligogenic trait, and therefore I can start looking at fewer individuals. I need perhaps 800 post-mortem brains to look at the correlation between methylation and disease. But that's a bidirectional correlation. It's not causation, because these methylation changes might be downstream of disease. Instead, with methylation QTLs, quantitative trait loci, I can start predicting how the genetics is influencing methylome, as we saw in the previous slide. And I can do that with fewer individuals of the order of hundreds, because this is a more oligogenic trait. But when I have that, I can do the following. I can predict methylation across a much larger cohort of individuals using this function here. And I can use the predicted methylation to then correlate these changes with disease. And because this is not all methylation, this is just the genetic component of methylation, which is predictable from genetics, I then have my causality back. These are genetic variants that are predictable from local genetic variation and that ultimately have an effect on disease, and therefore I get causality back. Moreover, the sample size is much larger than what I would have if I was looking at methylome-wide association studies, because people are much more generous with their blood than with their brain. They don't mind you taking a peek at their genetic variation, but they like to hold on to their brains until they're done using it. So using this, we can basically look at the combination of genetic variants that contribute to DNA methylation changes across dozens of loci and start predicting the new loci that are now causally associated with disease through multiple genetic variants that act upon them, even in loci that are not genome-wide significant. In this particular case, GWAS genome-wide significance is above the top of this figure. They would be at 5 times 10 to the minus 8 compared to only 10 to the minus 6, which is shown here. But instead, I find half a dozen loci in chromosome 6 alone that allows to now start inferring new genetic regions associated with obesity and specifically with Alzheimer's, and specifically the cell types and tissues in which they appear to be active. And I can do that across methylation, I can do that across transcription, I can do that across their combination to basically paint multiple pathways of action on the disease. So here's the result for Alzheimer's disease, looking at positively and negatively associated genes whose expression is either protective or inducing of Alzheimer's. And in the purple loci, we're basically starting to assign candidate driver genes and their direction of effect and their tissue of action. But in these gray loci, we're associating new regions altogether that were not previously suspected to play a role in Alzheimer's, giving us a new hint to potentially targetable loci. But again, everything I've showed you so far has been at the tissue level, not at the single cell level. But the brain is dramatically different between oligodendrocytes, astrocytes, microglia, neurons, excitatory inhibitory neurons, and so on and so forth. So we'd like to actually start understanding the individual cell type specific effects of this epigenomic and transcriptional variation. So to carry this out, we've now had a series of successful collaborations with Li Huitai, with Kevin Egan, with Veronique Belzil, with Brian and his colleagues, and with a large number of investigators across a large number of traits. For Alzheimer's, frontotemporal dementia, Lewy body dementia, ALS, Huntington's, AD with psychosis, schizophrenia, bipolar disorder, Down syndrome, autism, depression, suicide, PTSD, and aging. We've gathered cohorts of dozens of individuals. We've profiled millions of cells across all of the major cell types. And we've profiled RNA expression as well as DNA accessibility across, in some cases, multiple regions of the brain to basically understand what are the effects of disease and what are the effects of genetic variation in the function of each of the cell types, and what are the genes that are affected, what are the pathways that are affected in each of those. The results are striking. This past May 2019, with Li Huitai, we published the first of these studies looking at variation in Alzheimer's post-mortem brain across 80,000 cells across 48 individuals. What we're finding is that every single one of these individuals has a consistent gene expression pattern, enabling us to now distinguish the gross cell type annotation of these individuals. If you look here across the individual cells across these individuals, you see that every major cluster is captured across all individuals. But what's really striking is that the individuals that are asymptomatic, the non-AD individuals, the individuals that show mild cognitive impairment or advanced AD, show dramatically different patterns within these major cell types. If you look at excitatory neurons, they are split by our algorithms into two clusters, one of which is associated with Alzheimer's and one of which is not associated with Alzheimer's. And that's true not just of excitatory neurons, but of every major cell type. If we start partitioning these cells into each of these individual cell types, annotating these cell types, and then subclustering each of these cell types into completely unbiased gene expression clusters that are learned without any knowledge of whether these individuals are sick or not, AD or not, male or female, and so on and so forth. What we're finding is that there are major clusters that appear that effectively partition every major cell types into two groups, a disease-associated group and a non-disease associated group. So if you look at AD, you basically find that there's two classes of excitatory, two classes of inhibitory, two classes of astrocytes, two clusters of oligodendrocytes, and so on and so forth. If you look at inhibitory neurons, same thing. Astrocytes, same thing. Cognitive decline, same thing. Braak stages, amyloid. So it appears that disease is influencing every single major cell type with systems level changes that allow us to subcluster the cells into individual groups. One of the most striking aspects was that if you look at the subdivision between male and female individuals are dramatically enriched for cells that are associated with disease, but male individuals are dramatically enriched for cells that are not associated with disease. And this is true again across most of the major cell types. So what this indicates is that women might actually be more resilient to AD. Why? Because even though their expression changes are much worse off, we selected these individuals to be phenotypically matched both cognitively and at the pathology level. So it appears that women are sustaining a much stronger burden at the gene expression level, but are not exhibiting this at the phenotypic level, which is quite dramatic, especially given the stronger prevalence of Alzheimer's in female individuals. We could also start looking at what are the genes that increase in expression early in the disease and increase in expression late in the disease by comparing these three groups of individuals in a pairwise fashion. And what we find is that early changes are extremely cell type specific. Late changes are much more cell type common. And similar changes are found in the downregulated genes, which are more cell type common actually early on, but more cell type specific later on for the downregulated genes. And the increased genes late in the process appear to be much more associated with stress, whereas the increased genes early in the process give us, in fact, more insight into Alzheimer's. We find that the sex differences between AD and non-AD are, that the sex differences are dramatic before AD between men and women with 3,000 differentially expressed genes, but that they increase in AD with 6,000 differentially expressed genes between men and women. So that basically suggests that we should really be thinking of the cell type specific effects of Alzheimer's. And in particular, we're finding that myelination pathways are upregulated in men, but not upregulated in women in Alzheimer's disease. And indeed, when we look at the pathology, we find that women show much more white matter loss, indicating that perhaps white matter is a sex-specific pathway that should be addressed therapeutically, and that we should be thinking about sex-specific and cell type-specific therapeutic targeting approaches in Alzheimer's and also more broadly. We're also able to profile non-coding changes based on the accessibility at the single cell level. In this particular case, matching very nicely between different cell types and enabling us to link non-coding variants to likely target genes based on the correlation between accessibility changes in the non-coding genome and gene expression changes in matching cell types. Here's one example for each of the major cell types, where we're finding that the DNA accessibility is reduced in Alzheimer's disease for TMSB4X, just like the expression of this gene is reduced specifically in neurons. For astrocytes, we find that RLPB1 decreases in expression in AD, and indeed accessibility decreases. For microglia, ApoE increases in expression, and indeed accessibility increases. For oligodendrocytes, Manolis 7A is in fact increasing in expression in oligodendrocytes, and same for its accessibility, it's also increasing. And if you look across many genes, in this particular example in astrocytes, you find that you can start correlating the RNA changes shown here in the arrow on the x-axis with the DNA accessibility changes shown here again with the arrow on the y-axis, looking at many coordinated changes across many genes, but also examples where DNA accessibility is already there, but RNA expression changes only during the disease, perhaps in those primed regions, enabling us to find a general concordance between DNA accessibility changes and RNA changes, and enabling us to now start fine mapping genetic loci into the candidate individual nucleotides that are altered in the context of disease. Using the DNA sequence of these individuals, which can be inferred from the RNA sequencing of these single cell RNA profiles, we can actually start looking at somatic mutations that are only affecting a subset of the cells in these individuals and likely occurred either early in development or later in the clonality of expansion of individual replicating genes. When we do that, we basically find that excitatory and inhibitory neurons show fewer mutations than glial cells more generally, which makes sense because they are post-mitotic, whereas glial cells are continuing to divide and likely continuing to accumulate mutations. We're also finding a class of disease-associated neurons, which are in fact accumulating many more mutations than the other non-disease-associated neurons, indicating that perhaps we should be thinking of these mutations and even these phenotypes at the level of individual cells, which basically accumulate a lot of quote-unquote bad mutations and therefore leading to dysregulation in these individual cells. And the more cells accumulate these bad mutations in an individual, the more likely that individual is to cross the threshold of disease. We're finding a lot of differences in overall disease burden between AD and non-AD individuals in specific cell types and between male and female individuals. And we're also finding that the disease genes affected by these somatic mutations are in fact vastly different between excitatory neurons and oligodendrocytes and other cell types, enabling us to pinpoint individual processes such as vesicle trafficking, Golgi intermediate filament that show repeated mutations occurring in mosaicism. We're also finding that a lot of these hits are clonal across patients. So for example, you see that this individual here has a large number of mutations in many oligodendrocyte cells and many astrocyte cells, affecting the exact same gene, which is likely to play a key role in Alzheimer's. We've since scaled these methods dramatically, going from 48 individuals to thousands of individuals, many different regions, and many different disorders. One of the technological innovations that enables us to do that is multiplexing. We're now able to process many batches, many individuals in the same batch, enabling us to also correct a lot of potential batch effects across different conditions by processing individuals across different traits simultaneously in the same batch. We were able to look at hundreds of individuals, enabling us to predict now quantitative trait loci across these individuals and start inferring the specific variants that are affecting gene expression in specific cell types underlying a large number of different disorders. So for every single one of these loci associated with schizophrenia, we can actually ask, where are these genetic variants acting, what gene are they affecting, and what cell types are they primarily acting in? And we can do that across many different disorders. We've also started looking at different brain regions and profiling both RNA accessibility and DNA accessibility across 7 different regions of the brain in both Alzheimer's and non-Alzheimer's individuals in each of those cases. The results are quite striking. We're able to now start aligning these regions and these cells across these different brain regions. We're able to actually recognize sub-regional cellular resolution in the structures of the hippocampus, for example, and the striatum that are associated with individual classes of neurons that are now able to sort of form their own partitions. And we're able to now start dissecting a large number of these disorders. Where's that piece? So this is what happens when you do Zoom meetings with the kids at home. This is my little 3-year-old who doesn't actually realize that dad is giving a talk. So if you look at ALS and FTD, and luckily my 7-year-old and my 5-year-old are here to take her away. If you look at FTD and ALS, what you're basically seeing is that there are, again, dramatic changes that are happening between these disorders. So there's a lot of comorbidity of ALS and FTD. But what we're finding is that in oligodendrocytes, for example, you see these opposing effects where in disease, the cells from FTD individuals are moving in one direction and the cells from ALS individuals are moving in another direction. And this is also reflected in the pathways that are altered in disease. If you look, for example, in the motor cortex, you see this dramatic change in endocytosis and apoptosis and autophagy, regulation, differentiation, and signaling pathways that are downregulated in a large number of cell types with the individual genes shown here in ALS. By contrast, in FTD, we see a lot of upregulation, specifically in excitatory and inhibitory neurons, of these neurodegeneration-associated genes, these translation genes and transcription genes, and these energy and synaptic pathways. So again, there's a huge amount to be learned in each of these disorders based on these single cell changes. Here are some examples from schizophrenia. Again, you can see here that we can distinguish these dramatically different gene expression profiles of different classes of inhibitory neurons, different layers of excitatory neurons, and individual genes and individual pathways that are altered, along with individual cell types where these appear to be altered in. If you look at the genetic variants associated with schizophrenia, you're finding that they're localizing primarily in inhibitory neuron changes rather than excitatory neurons, and of course, not in other glial cell types. And if you look at the pathways, you see a lot of axon guidance, synaptic vesicle regulation of neural projection, a lot of pathways that make a lot of sense and that give us a handle for developing new therapeutics against these devastating disorders. If you look at Huntington's disease, you basically see that in control individuals, these indirect pathway spinal projection neurons in the striatum versus the direct pathway spinal projector neurons, the projection neurons, are in fact quite separate in their expression profiles. But in the context of Huntington's, they appear to be losing their identity and merging, again, consistent with what was expected from individual papers many years ago. And if you look at these genetic variants, as I mentioned earlier, they allow us to now go beyond just what are the gene expression changes that are happening, but also what are the genetic variants that are driving those in order to actually start painting paths of causality in these different disorders. We've also started deconvolving these profiles of thousands of individuals in bulk brain into the single cell profiles, and then using the two in combination to recognize cell type proportion changes and how these proportion changes are associated with disease. For example, with increased amyloid, you basically see a decrease in excitatory neurons, but an increase in astrocytes, and vice versa. With increased cognition, you see increased fractions of excitatory neurons, but decreased fractions of astrocytes in these deconvolved patterns across thousands of individuals. You can see that with the progression of Alzheimer's, there's a massive amount of neuronal loss. And with progression of age, there's also a massive amount of neuronal loss, and conversely, astrocyte gain again. We can also find genetic variants associated with changes in cell type proportion. In this particular case, we find cell fraction QTLs localizing in this TMEM106B associated with frontotemporal lobar degeneration, but not with Alzheimer's, even though the cell type proportion appear to be contributing to Alzheimer's disease. So individuals with protective variants have more excitatory neurons, and individuals with a risk variants have fewer excitatory neurons, perhaps contributing to the disease pathology. And the mechanism underlying this appears to be the loss of a CTCF binding site, which controls the formation of these topologically associated domains, which then leads to this topological change and an increase in TMEM106B levels, which leads to increased cytotoxicity. And again, as we saw earlier, we find that the vast majority of these genetic variants underlying Alzheimer's appear to be acting specifically in microglia, consistent with our previous findings. We've also started expanding this across multiple phenotypes. In the context of Alzheimer's, we know that neuritic plaques, neurofibrillary tangles, neuroinflammation have dramatically different signatures, even though they're all common underlying conditions of Alzheimer's disease. And if we look at the methylation changes that are associated with each of these signatures, we find that they actually impact different regions of the genome. So we can start partitioning the genome according to the combination of phenotypes that these regions are associated with. And indeed, this allows us to now start partitioning both promoter and enhancer regions in different subsets, which are enriched in different cell types of action. Embryonic stem cells versus immune cells versus fetal brain are associated with different combinations of these endophenotypes and also with different pathways. We've expanded these to electronic health records to basically start looking at the comorbidity patterns across many individuals and start learning common patient phenotypes and grouping these patients across all of these different indications, enabling us to now both distinguish patients more specifically, and also start completing the electronic health record based on imputation of these sparsely populated data types. And what we're finding is that the specific classes of patients that we're discovering are also associated with expression changes inferred from the genetics of these individuals using the GTEx cohort and then pinpointing specific cell types where these might be acting. We started looking at convergence of these pathways across many different tissues, profiling muscle and fat and gut and brain and liver and heart and blood across both mice and humans in weight loss and in exercise, and then finding how these gene expression changes are affecting not only the stromal cells, but also the immune cells, the resident immune cells of all of these different tissues. We're using this to use blood as a biomarker that predicts the effect of changes in inaccessible tissues. For example, in Alzheimer's disease, we're finding that blood samples are showing epigenomic changes, specifically in macrophages associated with AD, but not in control individuals. Looking at H3K27 profiling in Buffy code, we can now start partitioning it into the different cell types, and indeed we're finding a lot of changes in macrophages and neutrophils. We can use these changes to now start predicting who has Alzheimer's and who doesn't have Alzheimer's based on the blood test. And the predictive factors are in fact including genes that are known to be associated with Alzheimer's from their genetics. We're also using blood as a biomarker and also methylation changes in the brain as a biomarker to calculate biological age versus chronological age for these individuals. And we're finding that delta age as a variable allows us to now start predicting genetic variants that lead to faster or slower aging across different individuals. And of course, in cancer, exosomes are allowing us to now start predicting changes in inaccessible tissues, and I'll actually start deconvolving the tumor fractions to start interpreting differentially expressed genes observed directly from the exosomes of these cells. We're also able to to estimate the mutations from these exosomal RNAs using both cell-free DNA and tumor whole exome sequencing. And we're also able to see how these mutations are converging into common pathways in a large number of these genes in cancer. So for example, we're finding that different genes have more or fewer regulatory elements converging onto them, but mutations in different individuals are recurrently found in those pathways, leading to a consistent dysregulation of a small number of pathways underlying cancer. And we've gone in and validated several of these to basically show specific long-range contacts across chromosomes in some cases. We're also looking at longitudinal effects, in this case, in a particular case, in collaboration with Geneviève Bolland over at MGH, looking at the progression of cancer and immunotherapy across different individuals, and the recurrent mutations that are found in these tumors across multiple lineages coexisting in these individuals. Lastly, we've developed a large number of computational ways of predicting driver genes and a large number of experimental methods for dissecting the function of those genes, looking at synaptic activity in imaging studies in high throughput, looking at editing of individual loci, disruption of individual genes, or activation and inhibition of individual gene regulatory elements in iPS lines, which we then differentiate into neurons, microglia, astrocytes, oligodendrocytes. And here's an example of successfully manipulating the BIN1 gene associated with Alzheimer's using these epigenomic alterations. We're now conducting calcium signaling and imaging in 384 well plates to basically infer these activity changes in perturbations in high throughput. And we've also developed a large number of assays, including Hydra, that allows to now carry out 7 million enhancer tests in a single experiment, enabling us to now start fine mapping these gene regulatory regions into individual SNPs and individual conserved elements underlying these complex disorders. And in collaboration with Andrea Penning in the context of the Cure Alzheimer's Circuits Consortium, we're now including hundreds of these genetic variants associated with disease into the brains of mice in extremely multiplex ways to basically understand the effects of each of these individual variants using single-cell RNA sequencing on the transcriptome of individual cell types. So in summary, we've basically looked at epigenomic alterations across different cell types to predict disease-relevant tissues. We've combined genetic, epigenetic, and transcriptional variation in the context of disease at the single-cell level to understand how genetic effects are acting across multiple phenotypes, across multiple tissues, and in high-throughput dissection for these elements. This is a series of wonderful collaborations and very generous funding from the NIH and several foundations. Li Huitai is a very long-standing collaborator in Alzheimer's and many of the brain-related disorders. These are the folks who led the Epigenomic Roadmap project. Many of them have now started their own labs, so we're looking for new postdocs. This is a collaboration for the genetics of Alzheimer's, the immune drivers in AD, the high-throughput assays, electronic health records, and mosaicism. And I'll stop there and see if there's any time for questions. Thank you.
Manolis, thank you so much. That was clearly a tour de force, and I know that only scratches the surface of what you and your team are doing, so thank you so much. I'll start with a question, then open up to my colleagues. We have about 5 minutes for questions. At the beginning of your slides, you had distinguished the Alzheimer's diseases as being late onset. And then in your last set of slides, you just mentioned Alzheimer's disease. I was wondering if you could clarify in your later work that you presented, was it a combination of early and late onset? And if not, do you have any hypotheses on any differences that might exist between early onset and late onset Alzheimer's disease?
So thank you so much for your question, Brian, and thanks for the opportunity to present. So for Alzheimer's disease, what we're finding is that if you use Alzheimer's as a quantitative trait based on the age of onset, you actually have a lot more power to predict causal variants than if you just use Alzheimer's as a binary phenotype. So age of onset complements all these studies dramatically and gives actually a lot more power. If you look at very early onset Alzheimer's, then it's mostly due to strong effect rare variants rather than the common effect, the common variants which are generally of weaker effect, of course, with the exception of the ApoE locus. And you know, the pathways involved are sometimes converging, but the individual genes are very often different when you look at the perturbations in early onset. We're also looking at specifically the effect of the ApoE2, ApoE3 versus ApoE4 genotype, and we're now preparing a paper that's basically looking at the single cell changes across these gradients of susceptibility. And indeed, we're finding that the same pathways are altered, as you see from the hugely polygenic effects that we find when we look at cases versus controls.
Thank you. I'll open it up to anyone else who has questions.
Hello.
Manolis,
Yeah.
This is David.
Go for it.
A lot of us are kind of wondering to what extent you've kind of found the road to the Holy Grail, in the sense that we're looking at these rare patients with undiagnosed disorders, and yet most of the times we're not finding the answer when we look at the coding sequences or even splice regulatory regions, and the whole genome sequencing that we do has added relatively little in the way that we're currently analyzing the data in terms of finding diagnoses in these rare patients. I'm kind of wondering to what extent, how close are you to going from an individual patient to applying some of these methods to try to figure out what's the cause?
Well, that's exactly the reason why we're looking at mosaicism. Basically, there's so much unexplained variation. Some of it is no doubt environmental, and I expect environment to be by far the largest predictor. But in some cases, individuals respond dramatically different from each other, even in the same environment. And we believe that all of these mosaic mutations and somatic mutations might actually be at the root of some of the missing heritability, if you wish, to generalize the term, that even in the sort of whole genome sequencing of these individuals, we don't find it. I want to be cautious that, unfortunately, this is only a post-mortem approach to try to sort of retroactively explain what's going on by basically looking at these mosaic mutations and somatic mutations in the brain, the post-mortem brains of more afflicted or less afflicted individuals. The blood exosomes and circulating blood mutations that can be detected are not going to explain a vast amount of predictive power, unfortunately, because they capture only a very small fraction of those cells, and they're not quite very reliable in sort of predicting what are the somatic mutations that are happening in these inaccessible tissues. But I agree with you completely that this is very much the Holy Grail. We're, of course, not there yet, but as we start understanding this gradient from common to rare to personal to germline, de novo, I think the next stage after that is really somatic de novo and mosaic mutations, which are not even 100% penetrant in one individual. And yes, we do see that they correlate dramatically with phenotype at the single cell level. So I believe that this is actually what's going to be happening.
I think we probably have time for one more question. Any additional question from colleagues?
I hope my question doesn't sound naive. I want to first thank you for an amazing talk. Could you tell me a little bit more about your tissue acquisition? Do you work with existing registries? For example, Aorta, do you work with GenTAC? I was just thinking about how refined brain research is, regions of the brain, cell types. In contrast, when the aorta is listed as just the aorta, it's sort of ignoring all the different regions and potential cell types and differences, et cetera. Again, sorry if this sounds kind of naive, but.
No, on the contrary, you strike the nail on the head. Basically, there's a huge amount of variation as you start looking at different regions and different tissues. If you look at the GTEx cohort, for example, there's a huge amount of variation that we find in this bulk sampling of RNA across these thousands of individuals and 50 different tissues. And in some cases, it's basically where did the doctor cut that particular tissue? And did we include more of the vessel, or more of the lung, or more of the brown fat or white fat, and so on and so forth. So in the brain, we're basically finding that every region of the brain is ever so slightly different from every other region. And as you start looking, for example, into the hippocampus, you basically find that CA1, 2, 3, 4, and they actually show very different expression patterns. And in fact, from the single cell expression patterns, we can actually recognize the different expression signatures of cells that are basically sitting near each other. And even from the different layers of the cortex, we're basically finding that different neurons are in fact showing different expression profiles depending on how far they project down these cortical layers. So what's really phenomenal here is that there's this technological convergence between single cell profiling and more imaging and in situ sequencing, with images basically being able to sort of show multiple cells at a time with individual RNAs being highlighted and their expression pattern across the different cells. That's on one side, on the imaging side, sort of moving towards genetics. On the genetic side, we're basically looking at these single cell sequencing in transcriptional and spatially aware transcriptomics. So basically looking at slide-seq, for example, and other related technologies, and there's new kits now from 10x looking at spatial transcriptomics, enabling us to now take very, very small regions of the brain or of other tissues, such as the aorta, and basically asking what is the expression pattern of these tissues across them. And lastly, we're, and even in the heart, we're now looking at coronary artery disease and using spatial transcriptomics there to basically start understanding what are the changes that are happening in CAD, in the aorta, and in other sort of regions of the heart. And I think that as this convergence is happening, we're going to be building these references to basically understand how the single cell profiles enabling us to understand the spatial relationship, but that ultimately we can actually, we will find out that a lot of that can actually be inferred computationally. That basically the single cell expression patterns, if we understand the right markers of each of the different regions, layers, locations and cell types and substructural regions, then we will be able to predict a lot of it computationally. So the reason why I'm so excited is because these new technologies have this massive amount of untapped information. And as we start tapping it, we will be able to see a lot more of these subdivisions. I think your question is exactly spot on, and we're seeing a lot of what's coming ahead.
Manolis, I just want to thank you so much for joining us this morning, sharing with us all your knowledge. We really do appreciate it. And also, I think I speak on behalf of all of us in saying, please extend gratitude to your daughter for her cameo performances. They were fantastic as well.
Thank you so much, everyone.
Great. Have a great day.
Bye-bye, you too.